OsteoBites is a weekly osteosarcoma webinar and podcast presented by MIB agents. Today, we're talking with Dr. Richard Gorlick, Division Head, Department Chair, and Professor of Pediatrics at University of Texas MD Anderson Cancer Center, joined by Dr. Andy Livingston and Dr. Khazal. They'll be talking with us about correlative science leading to active and emerging clinical trials. So welcome to OsteoBites today, everyone. Thanks for being here. My OsteoBites of the day, a bowl of cherries, going to be delicious. We're really excited today to welcome the team at MD Anderson. We are so lucky we get Dr. Andy Livingston, Dr. Khazal, and Dr. Richard Gorlick. Dr. Gorlick is a two-time Spectre speaker. He is the Division Head, Department Chair, and Professor of Pediatrics at the University of Texas, MD Anderson Cancer Center, where he is also the Director of the Pediatric Sarcoma Research Lab. MIB agents are so grateful to him for working with us on projects like the Testing and Research Directory, which is an excellent tool if you have osteosarcoma, or even if you treat osteosarcoma. Really great. It's on our website. Please check it out. Dr. Gorlick and team are talking with us today about correlative science leading to active and emerging clinical trials, something we love to hear about. A great panel is part of the deal today as well. All of them are osteo warriors, including Charlotte Murdoff and MIB agent junior board member Ryan Kennington. And both Ryan and Charlotte are pursuing careers in medicine. Amanda Levine, our beloved patient advocate, is joining us as usual. And I'm your host, Ann Graham, president of MIB. MIB Agents makes it better, MIB, for kids with osteosarcoma. We help kids facing this aggressive cancer by providing research, education, direct patient support, and educational programs. If you're on this webinar, you know that osteosarcoma is the oldest known cancer with some of the oldest known treatments. So we also support the researcher and physician community with an annual conference through OsteoBites and by funding meaningful osteosarcoma-specific research. A mission of this size desires the hearts and hands of many. We welcome your participation as well to help us make it better. With that, Dr. Gorlick, would you begin by introducing yourself, please?
Yep. So thank you very much, Anne, for inviting me to sort of present at this OsteoBites. And I took the sort of liberty of sort of adding a team. I have my Doritos here, but I'm sorry, it's going to be hard to eat them through the mask. But I prioritize having my colleagues here rather than being able to eat the chips. And I'll eat the chips in about an hour. So as you mentioned, I am the Division Head of Pediatrics and Department Chair of Pediatrics at MD Anderson. Folks who are in Texas always sort of have this assumption from others that they've been there forever, but I am actually a lifetime New Yorker who's only been in Texas since 2016. You know, as mentioned, I'm a childhood cancer survivor, but my whole career has been focused on working on osteosarcoma. At the moment, what's kind of a little bit of an oddity is, you know, in MD Anderson, actually, osteosarcoma patients get cared for in both pediatrics and in cancer medicine, the sarcoma department. They are unified by the AYA program, which is co-led by Andy Livingston. But as another bridge between the two, at the moment, I am the interim chair of sarcoma in medical oncology, as well as the pediatric role. So we're a very unified team. I hope I'm exemplifying that by having members of the team here. I'm going to show when I present that the work is actually even of a greater group of people, but we have sort of limits on how many folks we can put in a room at a time and maintain social distancing. So this is the group that's here. So thank you.
Hi guys, good afternoon. My name is Andy Livingston. It's great to join you again. I was fortunate to be part of a prior session. So I am a pediatrician and also an adult oncologist. I focus on treating sarcomas and have a particular passion and interest for young people and osteosarcoma. So a lot of my research is looking at immunotherapies, but also developing new early phase clinical trials in osteosarcoma, some of the work I'll share with you guys today.
All right.
Hello, good afternoon. Thank you so much for the invitation. My name is Sajad Khazal, and I'm one of the pediatric stem cell transplantation and cellular therapy physicians. I specialize in immunotherapy and cancer diagnosis, both sarcomas and hematologic malignancies. We also do stem cell transplant for non-malignant disease. So I'll be happy to share with you one of the clinical trials that we have at MD Anderson. And thank you for the invitation again.
Hi, my name is Ryan. I was diagnosed with Osteosarcoma in 2014. I had a limb cell linking surgery, and then a year later, I had a BATS to get one lung metastasis removed, and ever since then, I've been cancer-free. Currently, I just graduated from University of Delaware with a degree in biology, and I hope to pursue a career in medicine.
Hi, I'm Charlotte. I'm a junior in high school. I was diagnosed in July 2018, and I had limb salvage surgery and finished treatment in March 2019. I relapsed in November and February, and I started a chemotherapy treatment in March, and I just finished in July.
Hi, I'm Amanda. I was diagnosed with OS in 1987. Since then, I went on to graduate college, got married, had 2 children, then was diagnosed with breast cancer. I survived that 10 years ago. 2 years ago, in 2018, I was diagnosed with ALS.
I did mention right at the start that this is the work of more than the people here. I've listed Najat Daw, John Gill, and Nancy Gordon is also contributing to this work. When patients are diagnosed with osteosarcoma, treatments broadly come in 2 categories. One category is standard of care type treatments, and the other big category of treatments is clinical trials. Standard of care treatments include a lot of things that are proven, and we all sort of agree are part of routine therapy. Some of those things include the very typical chemotherapy regimens we use to treat newly diagnosed patients, like regimens called MAP with methotrexate, cisplatin, and doxorubicin. Surgery is very much part of standard of care. Everybody with osteosarcoma needs surgery. When patients sometimes have osteosarcoma come back, there are drug treatments like ifosfamide, topicide, that some believe are proven enough that they can be given in some circumstances. There are some tyrosine kinase inhibitors that fall into that category. But at some point, once we get past the treatments that we know work, we get on to treatments that we're not sure work. Sometimes those type of treatments are offered in the form of clinical trials. They are offered in a way that we can sort of give it in a standardized manner and in a group of patients that we know we can describe so that we can use the information from every single patient to benefit everybody else. It's a way that we ensure generalizability. It's the way we know, like if we got a result that's sort of unfavorable, we don't keep doing it. If we get a result that's favorable, we can start moving it towards a standard of care. And a lot of doctors like Paul Myers, who was one of my mentors, will always talk about, therapies that are experimental that aren't really given in the context of clinical trials. And the problem with that is it's hard to interpret what that actually means. And even if it's given to a few patients, he'll say the plural of anecdotes isn't data. At the end of that, we don't know what to make of it. And so there are barriers to performing clinical trials. They take time, they take effort, they take expense. But I'm going to always advocate that clinical trials are the way to go because they advance the field in general. That's not to say that every patient or parent should agree to participate in a clinical trial. These are all research. Participation is always optional. And you have to understand, what's the price of participating in the clinical trial and make it as an informed decision that this is something that you want to do. I do think every family and every patient should be asking, though, at any point in time, what are the clinical trials that are available? Just knowing about them doesn't mean you have to participate, but information is always good. And that gives you an idea of the range of treatment options that are things that you should be considering. Meaning if there's no clinical trials and it's all standard of care, there's usually one path. If there's 10 clinical trials that are options, knowing about all of them gives you an idea of all the choices that are out there, and information is always power. So I wanted to start by mentioning that clinical trials come in a lot of forms. They come out of things like cooperative groups, they come out of institutions, they come out of industry. And a lot of my career was spent being actively engaged in the Children's Oncology group. Many folks associated with me with that group for a period of time because I led their bone tumor disease committee for 7 years, and I remain committed to that organization. I have a research laboratory, and some of the work from the research laboratory we directly send to the COG to use them to sort of define clinical trials. I'm not going to go through the busy parts of the slide, but I'm really using this to make the point that cooperative group trials are wonderful. I'm fully supportive of them. That said, I can't, in a short period of time, talk about everything. And I brought together my colleagues from MD Anderson because I want to focus on what's happening at MD Anderson. MD Anderson is a place where there's a lot of bench-to-bedside research, a lot of clinical trials opportunities, and I'm going to emphasize them. And the point of the introduction is standard of care exists, cooperative group trials exist. These are all good things, but I'm going to take my opportunity today to mostly talk about institutional trials. I'm going to start this off because this way I can sort of get my sort of opening soliloquy out before I have my colleagues talk about a couple of other trials. And I'm just going to talk about a couple of examples. And one of the examples is a phase 1/2 trial that we have of inhaled gemcitabine, which is a chemotherapy some of you may have heard about, for patients with metastatic osteosarcoma. This is a trial led by Sajad Khazal with Nancy Gordon providing a lot of the science that led to the study. So this work I am involved in, but this is not my work. So we all know that pulmonary metastases are a problem. We know gemcitabine is an active agent. We know, we can give drugs in an inhalation manner, and when we give them as inhalation, we sometimes get higher concentrations in the lung. And we know inhaled gemcitabine as a drug itself has been given in adults with lung cancer, and it's been given safely. So what Nancy Gordon's group did is studies involving mice and studies involving dogs, showing that inhalation gemcitabine using a metastatic osteosarcoma model improved the survival of the mice. They also showed that this treatment works in dogs with pulmonary metastasis. And basically, we know this works, like the top box is how the middle picture is kind of how mice get gemcitabine. Those are mice that have osteosarcoma. It shows how dogs get osteosarcoma, and then ultimately gets to, how people get how get the inhalation treatment. It's not how they get osteosarcoma. It's how they get treated for the osteosarcoma. Sorry, misspeaking. So it's basically always a nebulizer equipment that sort of works inside of a tent or inside of a cage or through a mask to allow people to know how to receive these things. The clinical trial, which sort of applies to human beings, has the following objectives. It's to define gemcitabine as safe, to describe its toxicity, to figure out how much is getting into them, and then once we get to the phase 2 component, looking at its efficacy. Again, every trial has eligibility criteria, and it's really critical that folks become aware of them. There are studies that will apply to some people and not others. Most of them are age parameters, disease parameters, but some are really around like organ function. Like if you're going to get a therapy that goes to your lungs, it's important that your lungs are working beforehand well to make sure that's going to go okay. And again, these criteria are usually created for a reason, and obviously they can always be discussed. But again, these are the type of information that help you define whether they may or may not apply. And again, the aerosolized gemcitabine is given every 28 days. People are put in a negative pressure room. The respiratory therapist comes with a tent. It's exposed. It's administered. And that's kind of how it's given. Patients can receive up to 12 cycles of treatment as long as there's no side effects and it's working. And again, there's all sorts of remote testing to allow spirometry remotely to make sure it's safe, see that it's not causing any harm. And really, just to sort of wrap up this part of it, this study is still at the phase where we're increasing the dose to see what's safe. Side effects still haven't been seen, so we keep escalating the dose. We're already on dose level 3, with that being the dose. And once we know, like the maximum dose or dose level 6 is safe, a series of patients will be treated at that dose to see whether this works or not. And again, this is just one option that'll be presented. Now, the advantage of having my colleagues is I could take a breath and sort of pass on to them. So for the next trial, we're going to hear from Dr. Livingston, I presume.
Great, thank you. So I want to tell you about, as you'll see, we have a lot of different clinical trials going on, but want to share with you about one of the clinical trials I developed, really how we brought what we were doing in the laboratory into the clinic. And so this was a really great opportunity. You heard already about some of Nancy Gordon's work, Dr. Gordon's work in the lab with gemcitabine. And based on that work, we started to think about how can we make that type of treatment work and work well for patients with osteosarcoma. So this study, what we're doing basically is taking some standard chemotherapies, gemcitabine and docetaxel, 2 IV chemos that have been around for a while, work pretty well in sarcomas, and thinking, how can we make them work better? And the way we're doing that is with something called hydroxychloroquine. So I will show you what that means. And that's probably a medicine you've heard quite a bit about recently in the context of COVID, which I won't talk about here. So this is a study I'm leading along with Nancy Gordon in the lab to really inform the science to think about how can we make this work and work better for patients. So what is this thing, autophagy? It's probably a word you've never heard before. I hadn't heard before. I started studying it, but it literally means self-eating. And so what happens to cells, like cancer cells, tumor cells, osteosarcoma cells, is when they're put under stress, there's different ways they can resist stress and resist chemotherapy. As they're being starved, getting radiation, getting chemotherapy, it stresses the cells, and they want to find a way to survive. So one of the ways cancer cells got smart about surviving is through this process called autophagy. And basically what happens is that the cell starts to eat itself or digest specific intracellular components to keep up its own supply of proteins, amino acids to build new proteins, or otherwise break down its own cellular components to maintain its energy. It's a way it can shut itself off from the environment. And if it does that, it means it's not taking in the chemotherapy and it's not dying. So we think that this thing called autophagy is actually one of the ways that different types of cancers, osteosarcoma included, can become resistant to standard chemotherapy. So there's been a lot of interest in looking at this in osteosarcoma, certainly out of our own group and our laboratories here at MD Anderson. I was fortunate to be involved in a few of these studies, but also there's publications nationally and internationally where people think, hey, this may be really important in osteosarcoma as a way to improve the ways that standard chemos work for patients. One of the things that I did early on after joining the lab was look at patients here at MD Anderson who had received standard chemotherapies, MAP-type chemotherapy, and try to figure out, do we see autophagy in the tumors, in the patient's tumors after they get chemo? And if so, are there other markers that might predict who will do well with this type of approach? And there's a lot of data on this slide, but basically the highlights are this. We looked at this thing called LC3B. We looked for autophagy, this stress marker, and we found that it was highest in patients after they got chemotherapy and very high, most even higher in the osteosarcoma metastasis. So we know it's there. We know it's happening. And then the second thing we looked at was this thing called HSP or HSP27. Could this be a predictive marker of which patients would have the best benefit from this type of treatment? And we found out that patients who have really high HSP27 in their tumor actually tended to do worse with standard therapy. So we said, well, maybe those are the patients we can help the most with this approach. So what we did was make a clinical trial, of course, and there's a couple different components. You know, the first part of the trial is to say, clearly we want to give patients this treatment and see how well does it work. We want to know, is this a safe way to treat patients? Because again, we're kind of taking standard chemo and adding to it with this pill called hydroxychloroquine. And then, perhaps most importantly, we're doing this study because we want to do better for patients with osteosarcoma. So can we improve how well the chemo works? And the way we're looking at that is, how well does the tumor respond? The second main goal or aim of this study is to try to figure out, can we predict which patients with osteosarcoma are the most likely to benefit from this unique type of treatment? So what Dr. Gordon's doing in the lab is taking patient samples, looking at a lot of these different markers, like this thing I was telling you, HSP27, and trying to see, does that marker predict which patients this treatment's going to work for? Because our hope is that we'll come up with a new treatment and be able to better predict who it will be most effective for. So here is another busy slide, but kind of showing you what we are doing on the study. Basically, any patient 12 years and up who has osteosarcoma that has come back or spread after standard chemo is potentially eligible. We have a long list of eligibility criteria as well that I'm not sharing here, but happy to discuss. Basically, patients will go on the study. What we'll do is find either a new biopsy or get an old piece of their tumor sample to look at some of the markers. You start on the treatment with the IV chemotherapy and hydroxychloroquine pills, and after every 2 cycles or every 6 weeks, we get new scans to see how well is the treatment working. If the treatment's working well and the patient's having few or manageable side effects, you keep going with the treatment for as much as a year. And what we're going to do is get a second tumor sample during the treatment to understand how well is the treatment working, what do we see changing in this autophagy stressors and HSPs that can help us to better understand how does this treatment work and how can we make better treatments for patients with osteosarcoma.
Okay, now we're going to move to Dr. Khazal.
Thank you so much. So I'm going to present a different clinical trial with a different approach. So here we're not using a chemotherapy or another modality. This strategy now is using live cells. So the cells that we're using in this clinical trial, we call it natural killer cells, which is an essential part of the immune system that can target the cancer cells. So few words about how we use the immune system. Either we can use the patient's own immune system by using some agents that can make the tumor cells visible or to the patient's own immune system, and that's why we call it checkpoint inhibitors. Basically, this type of medication, what we call it, releases the brake from the immune system and makes the patient's own immune system identify and target and destroy cancer cells, including osteosarcoma, versus an adoptive cell therapy strategy in which we take immune cells from different donors, and that's why we call it an allogeneic, which means a different person immune system, specifically the NK cells, and we use those cells and infuse it to the patient to target the cancer cells directly. So this clinical trial was based on the safety that we here at MD Anderson from our adult transplant colleagues, where they used NK cells from stem cell transplant donors to augment the chemotherapy and the radiation that we gave to adult patients with leukemia specifically, and adding this NK cell or natural killer cell cytoreductive strategy, which means an anti-tumor strategy, in addition to the chemotherapy and radiation before stem cell transplantation. And the data from the group who transplanted patients and used the NK cells showed that the NK cell infusions are safe, and this was also translated to improved outcome in terms of progression-free survival or the time that the patient survived leukemia-free. The major obstacle or the problem that was there from the beginning by using the NK cells is the small number of NK cells in the peripheral blood or from the donors. So the strategy that that we have at MD Anderson is the ability to expand those NK cells in the lab by almost 1,000-fold. That way we can really have an augmented anti-tumor effect from this type of treatment. This was also based on preclinical data, the fact that we're using NK cells for solid tumors. In vitro, when we put the cancer cells from Ewing sarcoma, osteosarcoma, rhabdomyosarcoma, neuroblastoma, we noticed that the NK cells, they have a direct anti-tumor effect. And also this was tested in vivo or in animal models, specifically mouse model. And as you can see from that figure, NK cell therapy or natural killer cell therapy alone or in combination with other treatments like irradiation in this case was associated with longer survival and the mice were cancer-free. So the pediatric solid tumor cell line is also killed by NK cells that is expanded by our methodology that is pioneered here at MD Anderson. The source of the NK cells that we are using in this clinical trial to treat patients with refractory osteosarcoma is a cord blood derived. It's an unrelated cord blood. We have our own cord blood bank at MD Anderson, which will make it much easier to have a potentially matched cord blood to the patient. We have to send an HLA typing on the patient. And we identify a potentially matched cord blood unit. The good thing about it is that there is no harm to donors. We don't have to have a volunteer adult donors. We collect the cord blood from donors in the delivery rooms at women, in the delivery rooms from women hospitals. And there's a low risk of something we call it graft versus host disease. Because NK cells are different from another type of immune cells, we call it the T cells, that can cause an immune reaction, which is not the case with the NK cells most of the time. This clinical trial is basically a phase 1 clinical trial, which means that we're trying to identify or to define if the NK cell infusion is safe in pediatric patients and young adults, and also to determine the maximum tolerated dose that can be escalated to phase 1, to phase 2, and future clinical studies. We're also planning to determine the persistence of these cord blood-derived NK cells in patients who are treated, and what is the anti-tumor activity of the NK cells. Patients, they have to have certain criteria. We call it inclusion criteria between the age of 1 and 40. They can have any type of pediatric solid tumor. And as Dr. Gorlick mentioned, on every clinical trial, we have strict inclusion and exclusion criteria, and we look for good organ function before we proceed. And also the patient has to have a partially HLA matched cord blood unit that we can isolate the NK cells from. The strategy how we treat the patients, once we identify a cord blood unit, we extract the NK cells from the cord blood unit, and it takes 14 days for the NK cell expansion to 1,000 folds in the lab. In the meanwhile, the patient will receive a combination of 2 agents called cyclophosphamide and etoposide. The purpose of it is to target the tumor first, the osteosarcoma, and also to serve another benefit, we call it lymphodepletion effect, to create a space for the NK cells to expand in the bloodstream of the patient so they can expand and benefit from the cytokines or other chemicals or stimulatory molecules in the bloodstream of the patient. And on day 0, we infuse the cells to the patient. So that's about it. Thank you.
Thank you. Thank you both for presenting. You know, what I wanted to just from here just talk about briefly is a couple of future directions. And really what exists at MD Anderson is a whole pipeline to develop drugs. One of the things we're very interested in is using proteins that are on the surface of osteosarcoma cells to serve as a means of targeting drugs more selectively to cells. You know, some of these trials are trials that, they made it to patients, but others, much more of them, are things that are sort of planned for the future. I know some folks must have said at one point to their doctor, like, are there any clinical trials that apply? And part of the reason I sort of want to make this point is, you know, you have to ask the question again and again. And I think it's not because the doctors are being deceptive. I think it's because things change over time. You know, today there are a certain number of trials available, and we presented a few that you can get today. But I think over time, I am an optimist. I think treatments will get better and better, and the options may get better and better. So you just have to keep asking about this. One of the targets that we're really excited about is this target, which is called CD276. This also has the name B7H3. There's actually a lot of groups all at once developing both drugs and cell therapy approaches to target this. There's, you know, cell therapy approaches being developed at other institutions. There's a CAR approach that's being developed specifically here. There's a number of companies that have drugs that target this particular antigen. A lot of groups are working. You know, we've shared this data with groups like COG who may pursue it. We're pursuing it locally. There's other groups that will pursue it as well. So again, the point is you have to ask the question again and again. I hope your doctors understand. You know, you can tell them I told you to do it. You know, and I hope they just understand that you want as much information as possible. I wanted to very briefly in closing talk a little bit about things that we haven't spoken about, as well as remind you about the things that we have, listing some of the clinical trials in osteosarcoma that are chemotherapy trials. The gemcitabine and docetaxel study with hydroxychloroquine is what Dr. Livingston presented. There is a phase 1 study of a liposomal irinotecan plus cyclophosphamide in pediatric solid tumors that's open nationally, that's being opened at MD Anderson. I talked about the phase 1 trial of aerosolized gemcitabine. That's an active trial. There's targeted therapies. So these are sort of approaches to use the genetics of the tumor to drive selection of patients for a particular therapy. There's a trial of olaparib, which is a particular DNA damage pathway targeted agent with ceralasertib. This is a concept that was developed at Dana-Farber and really is Katie Janeway's work. And part of the reason I was sort of highlighting this one is a lot of these trials are done at more than one place. That particular trial we're about to open at MD Anderson, we think it's a great concept. We'll have it available here as an option for patients who are seen here. Again, you know, we've talked about a lot of ways of trying to make sure people get to, you know, get as many opinions as possible. Understand a lot of us are friends and work together all the time. We're not as competitive as you think we are. Actually, we're one team with a common enemy of osteosarcoma. There's a study of regorafenib that's going to open at multiple sites nationally, adding it as a maintenance after first-line therapy. Again, this is something we're planning to open here. There's a PD-L1, now we're on to immunotherapy trials. We have a trial of nivolumab or nivolumab and azacitidine in patients with recurrent resectable osteo. That's soon to be opened at MD Anderson. And actually another institution-specific one, which is a phase 2 study of durvalumab and oleclumab. I'm going to kill the names on these. This is another Dr. Livingston study. We have a study of humanized monoclonal 3F8 with GM-CSF. in the treatment of recurrent osteosarcoma. This is a trial, I wanted to make it clear, we're not specific to Dana-Farber. This is a trial where this is a concept that was developed at Memorial Sloan Kettering Cancer Center, placed near and dear to my heart, having spent the first 11 years of my career there. That trial will be soon opened at MD Anderson as well. Phase 1-2 study of an AXL targeting antibody drug conjugate. This is already open at MD Anderson, open at other places. There's a study of, I didn't talk at all about tumor infiltrating lymphocytes. That would have been another talk. We actually have 2 trials here of that concept. This is open at MD Anderson. The industry-sponsored one is open at other sites, and then there's a MD Anderson-specific TIL study. And the allogeneic NK cell trial is the trial that Dr. Khazal just discussed. I am not talking about things that I think are about a year on the horizon. I think next year the list will be larger. These are things that are all eminently opening, meaning months, not years. I think there's much more coming. And I think for osteosarcoma in general, just sort of in closing, I think, you know, there's a lot of pessimism because there were years without progress. But I'm an optimist. I think, you know, with all these new drugs coming on board, I am optimistic that we will finally make progress in this disease. We're all on the same side, and that's what we all want. And we have new tools that we never had before, so I don't think the past lack of success is really going to hold us back. I think we are going to get where we want to go. My last slide, there's a lot of people who make everything I do happen. Actually, I do very little of anything. So obviously, you know, I have a tremendous number of clinical colleagues. You know, I have lab folks, collaborators, et cetera, et cetera. Obviously, we get funding from multiple sources. And really, you know, my final thanks is, you know, after listening to me speak at multiple MIB factors, I'm sort of shocked that you guys want me to give a webinar. But I thank you guys for continuing to listen to me and encouraging me to present. You know, the sentiment in the heart's there, even if the delivery isn't perfect. And obviously, thank you guys for all you do at MIB for making it better. We really appreciate it.
And I have to tell you, after this presentation, my cup of joy is overflowing. Like, I can see it, Amanda, too, right? Amanda, are you not like, yeah. This is so much more than we anticipated hearing about today. This is a lot of clinical trials in place and so many coming up. And I think you addressed this a little bit. There's never been this much available for osteosarcoma, like in the history, in 77 million years since the dinosaur that was just revealed as having had osteosarcoma. We've never had this much research. Why is that? How is this happening?
Yeah, you know, I think that's a great starting comment. And, you know, I think we had several decades without progress. And I think during those decades, we really had 5 drugs that worked. And a lot of the trials were manipulating the 5 drugs in different ways. And at the end of the day, you're going to get what you can from those 5 drugs. And I don't think you're going to get much more out of them. I think really what changed the world is really the revolution in molecular biology and the lab side of it. You know, as a cancer survivor, I came to pediatric oncology with no commitment at all to laboratory research. Actually, famously, before I did my training, I never spent a minute in the lab, and I envy Ryan, who sort of has taken the right path and has done research on his way to a potential medical profession. It wasn't how I thought about things, and it was really inspired mentors at Memorial Sloan Kettering who really said, you help one patient at a time through being a doctor that takes care of patients, but you're going to change the field through research. And I actually bought into it. and sort of made a very big part of my career research, along with many others, sort of taking a similar direction. And I think it's the explosion in understanding the genetics, understanding the immune system, being able to produce antibodies readily to target things, being able to produce NK cells to target things, being able to genetically manipulate NK cells to, target what you want them to target, coupled to the ability to rapidly make drugs. An antibody drug conjugate is really three parts. It's an antibody that binds a protein, a linker, and then a drug. And so those can be mixed and matched in so many different ways with common constituents. It just opens the door to things that didn't exist before. And I don't remember there being, 2 trials in osteosarcoma at a time, let alone, 10 or 20. So it's an exciting time. It really is.
If a patient wanted to consider a trial, what is considered for travel to be eligible? Can a patient be in an MD Anderson trial but get the treatment at their local hospital? And if no, is travel back and forth allowed? Or would it require staying in the MD Anderson area?
In general, like a trial is only open at selected sites. And you have to go one of the sites it's open to at least get started. It depends on the specific of the treatment for their trial, how long they have to be by that site to sort of get treated on that particular trial. There are many trials where you can come in once every couple of weeks and then do everything else locally. There's trials where, the treatment's a little more intense and you kind of have to be local more often. I think part of the reason we collaborate with so many people is, just to try to help with geography. It's not an accident that, us in Texas do some of our trials with folks in the East Coast, so that there's sort of an East Coast and a Southern option. A lot of times we throw in a site on the West Coast so that there's sort of places to get it multiple parts in the country to try to minimize travel. I don't know. Your trial, I think they stay on. But your trial, they may come in episodically. So we have an example of both.
Right. For the NK trial, for example, the patient needs to be here during the screening and the chemotherapy and the NK cell infusion. Actually, after that, for 2 weeks, we have to see the patient twice a week in the clinic. But then after that, can do the labs locally, and we can follow up remotely by doing virtual visits. And then at a certain time point, which is a month after, we need to do the scan, and it has to be at MD Anderson. So it's really, as Dr. Gorlick mentioned, it's trial dependent.
Yeah. And the allogeneic NK trial is a big treatment, but kind of all at once. So it's sort of designed to come, get it, and then whatever, whereas other treatments like the inhalation chemotherapy are much more episodic. Longer periods of time, less intense. We don't know which is really right, actually. Do you have any other?
Yeah, I would agree and echo those points just to say that, every trial is different. Certainly as we're thinking about people as designing these trials, leading these trials, certainly doing things that can be done closer to home, things that can be done safely, allow patients to travel back and forth, that is a big consideration, but also wanting to do that in a safe way. So I think, if you're ever considering a clinical trial, those are the right type of questions to ask, but of course every study is a little bit different.
And we all answer emails. So if you have questions, just reach out to the person who leads the trial. They'll always answer.
First, I want to thank you so much, Christy, giving so much hope to so many. First, all these trials are clinical trials that go. Also, are there any placebos, or does everyone in the trial get the drug? And you mentioned that some are other locations, but the ones that you spoke about, are they other places than
clinicaltrials.gov is a great resource, and actually all of these trials are listed on clinicaltrials.gov. Most of us in the field want to list it, meaning it's a great resource if people want to use it. The biggest problem I have with clinicaltrials.gov, and I joke with Anne at the start of this that I don't even know what YouTube is when I go to clinicaltrials.gov, I find it hard to find anything, actually. It's actually kind of hard to search. And, that's kind of why I say use your doctor to some extent to help guide you what's out there. I have trouble using it. If you're more information savvy and you don't, that's wonderful. It's a great resource. None of these trials are placebo-controlled. Actually, in the context of a disease that's kind of life-threatening, not kind of life-threatening, is life-threatening, I think the ethics of giving somebody something just to sort of make the experiment better is probably borderline ethical. I don't know that I would ever be willing to support, like at MD Anderson, somebody doing a placebo-controlled trial in osteosarcoma. In certain contexts, it's wonderful. Understand, vaccine trials, things like that. I'm not commenting on that broadly, but not in osteosarcoma.
Is there a charge for the patient?
For the trials, the clinical trials, there isn't for participating relative to research, but insurance is billed for what's standard of care. So, you become a patient of MD Anderson, and so we can't promise there won't be some billing related to being just an MD Anderson patient. There is never added charges because you're on a clinical trial. That's always covered by the research aspect of it.
Between Amanda's group and MIB agents, we connect regularly with almost 10,000 osteosarcoma patient families. Many times we get the question, I'm metastatic, what now? What do I do now? Mm-hmm. And just like you pointed out, going to clinicaltrials.gov that should be the first line, but that is so technical in many cases and not simple is a real barrier to the patient, of course, for seeking the treatment, and to the PI for recruiting for their trial. So there has to be a better way, and we're as a community, as you said, our common beast is osteosarcoma, and we all work together in the meantime. So, for example, Osteosarcoma Institute has their helpline, which is so brilliant. It's manned by an intake person who hands the question over to Dr. Lee Hellman, who's an expert in osteosarcoma. And I think he can help in this area to point people in the right direction. In addition to / apart from that, is it fair to say that for us, who neither one of us give medical advice to anybody except call osteosarcoma, the osteosarcoma hotline, or go to a sarcoma center immediately? Those are the two pieces of advice we do offer. Would it be fair to say to a patient, hey, call MD Anderson, tell them where you're at in your treatment, and see what they have available?
We, like many large places, are a huge sarcoma center. MD Anderson's the biggest cancer center in the United States, and obviously the sarcoma program is huge. That said, I love my colleagues. So, Lee Hellman's great, Katie Janeway's great, Paul Myers is great, Pete Anderson. Everybody is going to love the particular provider they've worked with, and please, pardon me if I didn't mention yours. I'm just listing folks at the top of my list. I do think all of the larger sarcoma centers are very much aware of what each other is doing, and we keep in touch with each other. I would always say talk to two so that you make sure, the one person you didn't speak to, the one person you spoke to didn't forget to mention something. But then from there, I do think we remain connected. Again, I am somebody who tends to tends to become philosophical at points in time. And Katie Janeway and I have had this long-standing conversation about one of the things that would be a great idea if we could ever work it out, and this was before COVID, which with COVID probably preventing any of this, is maybe we should have a once-a-month clinic that sort of rotates through, 10 of the centers, where we sort of all go together and sort of you get to see 3-4 of us at once. To be honest, it would facilitate your guys' life because you could get a Memorial Boston, MD Anderson opinion all at once. Obviously because of travel now, things are more complicated, but I actually think it's something we should move towards. We're all working together. There's only one enemy, osteosarcoma.
Yep, agreed. So I want to add my vote for that, the Dream Team Clinic. If we can help facilitate that, Amanda and I would bring the patients to you. Okay, so totally on board for that. Brian, you're...
All right, so I kind of have a quick question to go off of what was previously, and do you think there should be some central resource the average patient should be able to go to just to see all the top clinical trials for each sarcoma center?
Yeah, I actually think, it wouldn't be that hard, and it's probably just worth the effort to gather a list or procure a list. Meaning just ask all, the 20, 30 people you want to reach out to, what are the trials that you think are available in osteo?
Actually, a couple of questions for Dr. Khazal. First one is, can you clarify the difference between NK cells and the killer CAR T cells? I hear kind of both used in similar contexts, and be interested to know what the differences are. Second question is, given the results that you saw in the mouse models, are you planning to test NK cells alone or in conjunction with radiation?
Very good questions. Thank you so much. Regarding the CAR T, so the CAR T is a genetically modified T cell. This is the very first FDA-approved gene therapy that was actually approved exactly August 31st, 2017, for the treatment of children and young adults with leukemia. And then a couple of months later, in October 2017, that was approved for adults with lymphoma. So the CAR concept is basically a genetic modification of a certain type of immune cells called the T cells. The NK cells that I discussed on the current clinical trial for osteosarcoma is not genetically modified. But can we do that? Yes. Recently, our group at MD Anderson published the results of NK CAR. So it's a genetically modified NK cell that targets leukemia and lymphoma. And there will be incoming work and emerging data about NK CAR for solid tumors for certain targets. So really, the NK cells that we have for this clinical trial is not genetically modified. It's simply expanded from a cord blood unit, and we infuse it to the patient. And the second question, we are testing it at the moment alone. The chemotherapy that we are giving it up front, it serves mainly as a lymphodepleting chemotherapy, which is it creates a space for the NK cells to expand in the bloodstream of the patient. We are not combining it with chemotherapy, as I mentioned, with the adult clinical trial that they use this NK cell in combination with chemo for leukemia. But that's the concept for future clinical trials. As of now, we're trying to determine and conclude that it is safe to give NK cells in children, and subsequent trials will definitely may include multiple infusions, genetically modified NK cells, or in combination with chemotherapy and radiation. So that's actually the future of the immunotherapy.
Yeah. So these cells are actually very different because it's the cell type is an NK cell rather than a T cell. When you put in a CAR, you're targeting it to a specific protein. These aren't targeted to a specific protein. But most of the time when you make CAR T cells, you're making them using their own cells. what the NK trial does is takes the cells from another person. They're allogeneic. They're not autologous. So they're not from the person going back into the person. And that's part of trying to enhance a recognition of the tumor as being more different. And it's a way of trying to get past the immune tolerance issue.
For the immunotherapy trials, do you need whole genome sequencing or RNA sequencing results prior to being enrolled?
Yeah, what's kind of interesting is actually the immunotherapy trials tend to be based on more broadly targeting osteosarcoma and really manipulating the person's immune system. So it's less about how the tumor looks and really how the sort of host looks. So the answer is no. Actually, there are a lot of doctors who try, who are interested in this personalized medicine approach. And what it's sort of understanding is that osteosarcoma can have a lot of different genetic derangements, and then trying to give drugs that target those. Some of the trials that I mentioned are in that personalized medicine category, like the ATR inhibitors. Those tend to be the trials where you have to have in your tumor the genetic change that leads you to go on to that study. Immunotherapy tends to apply to everybody with osteo. The personalized medicine trials are the ones where the genomics of the tumor matter more. I do think a big part of our field is trying, though, independent of that discussion, figuring out why somebody responds or doesn't respond. I presented at MIB, a laboratory project looking at drug resistance, and some of it really is getting at, we give the same drug to, 100 people, and some people it takes care of it, and some people it doesn't, and, we don't know why. And that's disturbing, beyond disturbing. So we got to figure out better why our drugs work and don't work and use them more smartly. But, it's mostly those personalized medicine trials that are starting to take advantage of the genetics to understand it. Do I encourage, this comes back to a related question, should everybody at some point get their osteosarcoma sequenced? I am an advocate of that. I think information is always good. Every once in a while it'll point a way to a specific treatment, and I think that's a good idea. I encourage those. I encourage donating tumor samples for research. I'm supportive of a lot of those things, but just didn't have time to roll them into one talk.
I have a question here that says, I noticed that many clinical trials have a lower age of 12 years. Is there a reason that a child younger than 12 years would not be eligible?
Yeah, that's a fantastic question. So in the olden days, the FDA, so the body that regulates drugs, considered trials that were in pediatrics, folks that were lower than 18, and trials that were in adults older than 18. And what they kind of required is before you go ahead and do testing in anyone below 18, you got to redo the safety studies after they've been done in adults to reestablish the dose in kids. What doctors like EYA doctors, and many of us have been advocates for this, are adolescents actually physiologically aren't different from adults. And we shouldn't need to wait to do a pediatric phase 1 trial before we tested in adolescents. The FDA over time has heard that. So most of the time, the age boundary of a trial is in 12 and above, is because a safety study has never been done in younger children. And so the 12 allowed it to happen before that happened. Why do you have to do studies in children below 12? Like pre-puberty, there's a lot of growth going on. And you got to realize that a lot of our drugs target growth. And so there is a reason to believe that growing children on the early age of the spectrum may have side effects that are different than adults. But why should we wait in teenagers to have to wait until those safety studies are done in babies and young children to get them in? So 12 is the FDA line for a lower age limit for a trial that hasn't had a pediatric safety study. From there, what I would say as an osteo, the reason we don't push for that ped safety study is even though there are some kids below 12, most are older. It captures most osteosarcoma patients. You know, there are occasionally patients 8, 9, 10, but, you know, babies never get osteosarcoma.
These trials only for recurrences, or are they also for primary diagnoses?
Yeah, these are trials for patients who either have recurrent disease or after treatment never got rid of it. You know, part of the reason, you know, you don't modify therapy so much up front is because we know, like, MAP therapy works. So you don't want to not give the standard of care drugs. You know, standard of care is always appropriate. So please, everybody, you know, with newly diagnosed osteosarcoma needs chemotherapy, needs surgery. Please don't skip the surgery. You can't cure osteosarcoma without removing it. Now, this was an immense amount of fun. Thank you very much for letting us go to talk to you guys. Great questions. Obviously, great level of engagement. This was really a lot of fun.
Telling you that on next week's OsteoBites, we will have another childhood cancer survivor as a speaker. It is an all osteo warrior everything when Dr. Kurt Weiss joins us. Dr. Weiss is a clinician scientist, a fellow for innovative cancer research, associate professor at the University of Pittsburgh School of Medicine, Department of Orthopedic Surgery, Musculoskeletal Oncology. He's the vice chair of translational research and director of Musculoskeletal Oncology Laboratory. Finally, Ryan would be modeling our most comfortable t-shirt ever made. Let's have it, Ryan. Yeah.
Oh, wow.
There it is. You can get one too, along with a fabulous cape, which, you know, is mostly reserved for superheroes and MIB agents. And you can get that when you sign up to Outrun Osteosarcoma. This is an important project for us. It's how we fund our research, our programs, and everything we do. So you can sign up on our website at info@mibagents.org and go under what we do. You'll find it there. We'd love it if you asked your friends and family to sign up too in honor of an osteo warrior. Your participation, again, allows us to keep trying to make it better for osteosarcoma families everywhere. Thank you for joining us today. And of course, thanks to our guests, Dr. Gorlick, Dr. Livingston, Dr. Khazal, our panelists, Charlotte, yay, Ryan, and Amanda. Stay safe, everyone. If MIB agents can be of help to you, please just say the word. Together, we make it better for osteosarcoma kids everywhere. Upcoming OsteoBite sessions are featuring osteosarcoma experts Dr. Kurt Weiss, Dr. Damon Reed, and Dr. Brian Flesner. We hope you'll join us. Until next Thursday, please use your powers for good for kids with osteosarcoma. Visit our website at mibagents.org.